Hello, and welcome to the Gene Lab for High School Worms in Space recording. I'll start the presentation by telling you a bit about why I study things in space, explain a bit about why worms in space, explain a bit about how we do experiments with worms in space, and then talk about results that we've obtained with worms in space. I'll then close with some more complicated material involving how do we take results from worms to understand things better in people. When we think about spaceflight, it's easy to get excited. It's a mystical place, not easy to conceptualize from your everyday experience, and there's lots of sci-fi movies about. Now that said, even though it's hard to conceptualize being in space, humans have been living and working in space for 60 years. As a result, we know quite a bit about what happens to the body in space. For example, we know that there are changes in the mitochondria. We know that there are shifts in the microbiome. We know that there are epigenetic changes. And we know that there are certain hazards associated with spaceflight which cause some of these changes. For example, altered gravity, altered radiation, and living in a closed and hostile environment. Now that one may seem a bit strange, but if you think about going to, say, college and being placed in a room with people you've never been with before, you realize that you will exchange germs with those people, and that basically is quite a different experience to being on your own. Now, when we think about studying biological changes in space, obviously one way to do this is to just concentrate on astronauts. If we're mainly focused on sending people to Mars, then we need to understand how do we keep them healthy and happy and safe on such a journey. However, there are some problems with studying astronauts, such as there aren't a lot of them, which makes it difficult to do large-scale studies and therefore gain large amounts of information just from studying astronauts. So just like on Earth, when we want to study people, sometimes we turn to model organisms such as rats or mice or fish or flies or worms, in some cases even bacteria and plants. And so there are pros and cons of working with different species. In the case of the worm, they're very small. It's easy to study hundreds of thousands of worms on one mission as compared to, say, 6 astronauts. So what's this worm look like? There's an example of one. They're only about 1 millimeter long. So they are microscopic, but despite this, if you noticed in the video, you could actually see that there's different stuff inside that worm, and that different stuff, just like inside us, is different organ systems. So for example, an alimentary tract, a reproductive tract, and muscles. So they're a microscopic multicellular animal, and because they're small, and as I said, we can grow lots of them, they're a pretty cheap and easy model for doing studies of molecular biology, or particularly molecular evolution, both on Earth and beyond Earth. So as I've said, they're microscopic, but they have multiple organ systems. That means that they're good for studying certain systems such as muscle. Their development is sensitive to the environment. That's important for 2 reasons. One, it means that we can use them to study different environments. For example, going to space, going to the moon, going to Mars. But it also means that we can manipulate their life history by placing them into stasis, so they can go into hibernation as an L1 or a dower, which means that we could potentially keep them in stasis for 6 months on a rocket for, say, doing a mission to the moon or Mars. From a more scientific perspective, other nematodes are found in extreme environments, such as the Arctic, the Antarctic, in the ocean at depths of at least 30,000 feet, in soil at depths of at least 17 feet, in the mountain soil in altitudes of greater than 14,000 feet, and they've been revived from permafrost after more than 40,000 years. So they're quite a diverse animal. More importantly, from an evolutionary perspective, reproducible mutations in response to laboratory-imposed environmental conditions are now known, and they can be observed in less than 3 years. This means that we could potentially take worms, make a colony of them on the moon, let them accumulate mutations to see how they evolve to living there, and then bring them back to understand how that adaptation beyond Earth is going. Now, in terms of human health, as I said, worms have muscles, so we can think about them as a model for human muscle. The worm has approximately 100 striated body wall muscles, which are functionally equivalent to skeletal muscle in vertebrates. And they connect the exoskeleton to allow for movement, just like our muscles connect to our endoskeleton to allow for movement. Notable discoveries using worms as they relate to muscle include we've uncovered the way that muscle gene expression is controlled, and this has led to the discovery of myogenic transcription factors such as MyoD, and serendipitously the discovery of RNAi. We've also learned that the control of muscle development has led to the discovery of evolutionary conserved sarcomere assembly. So what does that mean? That means the way that our muscles get put together are conserved from worms all the way up to people, and those are predominantly based upon external mechanical forces to the muscle, not internal forces within the muscle. We've also discovered the role of thousands of individual genes on muscle development and health. And from a human disease perspective, the worm happens to have the same gene called dystrophin that is known to cause muscular dystrophy in humans, and therefore we can study novel treatments for muscular dystrophy using the worm C. elegans. So from a practical perspective, one of the reasons for using worms is they grow quick. Their generation is 3 to 4 days or 7 to 10 days for defined medium. That means it's fairly easy to do experiments in a short period of time. Logistically, they only need an incubator and worm food, so they're not particularly costly. And again, they're small, so they don't take up much space. You can use most techniques as in other species. This includes PCR, immunofluorescence, transgenics. I should point out that the use of transgenics, including GFP, resulted in a Nobel Prize for studies in the worm. You can do westerns, southerns, microarrays, RNAi, which again was discovered in the worm and again resulted in a Nobel Prize for studies in the worm. You can use drugs, you can use inhibitors, you can use CRISPR, and then the worm has an essentially invariant cell lineage, which led to the discoveries of apoptosis and microRNAs, both of which also resulted in Nobel Prizes for studies in worms. Now, I said that the life cycle is sensitive to the environment, and we do know that the alternative Dauer state is controlled by the insulin and TGF-beta pathways, both important pathways in mammals. Now, obviously, worms are not people. They don't have eyes, they don't have arms, they don't have legs. So we know they're not the same. And some of the other limitations are that they don't have an inflammatory system, they have a poorly developed immune system, and they have no satellite cells. So it is not possible to use the worm to study everything that you might like to understand about an astronaut using the worm. So to close this portion of the talk, the worm is a well-defined genetic model organism with basic physiologic systems. It's the simplest lab animal for studying neuromuscular function in vivo. Thus, one strategy for translational research is to look first in worms and test extension into humans. I'll talk about that at the end of the talk. A basic premise that I use for this is the idea that mutations in insulin signaling promote longevity in worms, rodents, and are found in people who live to be more than 100. All right, a history of worms in space. As you can see from this chart, worms have been going to space for more than 30 years. Now, if we look at the different experiments, we can see that they're mostly short duration. There is one experiment where they went for 6 months. There's another experiment where they went for about 60 days. So you can see the worm is a versatile model for both short and long duration experiments in space. So how do we do an experiment with worms? If we want to do a simple experiment, we can put worms in their food inside plastic bags. If you look to the left, you'll see little bags that look like duck sauce packets. These are about the size of a ketchup packet, a soy sauce packet, or a taco packet. And effectively, we put these plastic bags, which contain the worms and the food, inside an experiment container, which is basically the size of an old flip phone. That flip phone can then be placed inside an incubator, and then the experiment can be initiated. In order to get it to the space station, we can send it up either with passive or active cooling, and that can be done in soft stow if we don't require active cooling. Once on station, we'll want active incubation, and then for return, if we deactivate the experiment in the freezer, we just bring them back either with passive or active temperature control just to ensure that they actually remain frozen. This type of experiment requires sample return and is a prime example of an experiment that you could use for doing gene expression studies in space. Here's an example of a more complex experiment. If you look in the upper left, you'll see little plastic cassettes, and you'll see that they have little cutout windows that are transparent in the middle of them. These are effectively cell culture cassettes. By putting the worms and the food in these cassettes, or putting the worms and food in one cassette and food in another cassette, what we can do is shown in this image to the right is we can link these cassettes together via tubing, and we can pump worms from one cassette to another cassette, say on a monthly basis, to make sure they remain fed. By coupling this with a video system as shown on the far right, we can actually watch the health of the worms over 6 to 12 months on the space station. In order to get this experiment to the station, again, they can go up with passive or active cooling, but once they actually are on station, they'll require active cooling and active imaging. Now, because we're imaging them on orbit, we can either return the samples, say for gene expression, or we cannot return the samples because we're just interested in the imaging data. So this is an example of an experiment where sample return is optional. So what have we learned from these experiments with worms in space? Key early findings confirm that worms grow normally and develop normally in space, that they have a normal ability to remove dead cells. This is important because that particular pathway is activated by radiation. Normal rates of mutation were detected. Now, this is in the case of worms being inside the shielded environment of the space station. If you place the worms outside the shielded environment of the space station, they do indeed experience higher rates of mutation. Some worms did experience movement defects, so there was individual variability in the response to space flight. When we look at the changes in the expression of the genome, up to 10% of the genome changes in response to flight. That's a fairly significant fraction. Key genes that change in expression include muscle genes and metabolic genes. These gene expression changes were followed up on subsequent flights, and they were also confirmed to occur at the level of the protein using proteomics. Now, a recent study looking at all of the studies that have done transcriptomics in space has looked for commonalities in the response to spaceflight. As we can see from this figure, metabolism and cytoskeletal changes are common. Changes in genes controlled by insulin and TGF-beta are common. Changes in genes controlling dopamine metabolism are common. But there are a limited number of microRNAs that change in response to space. Now, the gene changes that we see in the worm do seem to be conserved across species. And this is an area that I will talk about a bit more. Okay, let's talk about some more complicated experiments once we have results from gene expression changes in space. So let's say that we find a bunch of genes that change in worms. For example, in this case, cytoskeletal genes that control the attachment of muscles to the extracellular matrix. Can we find the same genes in humans? The answer to that is yes. So what happens then if we perturb the function of these genes in worms on Earth? As shown in green, normally the worms have nice straight sarcomeres, straight lines. But when we disrupt members of these attachment complexes, you can see that these straight lines basically dissolve into blobs. And with the graph in the middle, you can see a black line where strength tends to be fairly evenly distributed across the spectrum, whereas when we mutate particular genes, as shown in red and blue, strength dramatically decreases as shown by the shift to the left. Now, if we do the same thing in people, we can see that strength declines dramatically in both young and old people when these attachment complexes are disrupted. And we can see in the top picture that the attachment complex proteins are usually at the periphery of the muscle prior to disruption. Following disruption, they move to the inside of the muscle. For these reasons, we moved on to test whether muscle strength declines in flight, as you might predict from these results on the ground. But we also found that when we disrupt these genes in both worms and people, mitochondrial function goes down, which means that both worms and people are less able to produce energy once these attachment complexes are disrupted on Earth. So this says that changes in muscle attachment complexes correlate with muscle function and metabolism, and they could potentially be causing some of the deficits that we see in flight. In terms of testing muscle strength using the worm, this was a little bit complicated. We had to develop a microfluidic device for measuring strength in the worms. And so as shown in panel A, we've got multiple chambers that we can place individual worms in. And as shown in panel F, we do that by using a microfluidic system to feed the worms into these chambers. And then as shown in B, we can actually see when the worms are in the chambers. And as shown on the top bit, we can actually watch as the worms deflect the pillars. Now we can think of these pillars as effectively being force plates. So the more force the worm produces, the more it can deflect the force plate, just like when people do exercise. So using this microfluidics device, we found, as shown in the graph, that worms are roughly 15% weaker in space, and that worms with a mutation in dystrophin are even weaker than wild-type worms when exposed to space, meaning that mutation in dystrophin does seem to predispose them for additional negative consequences of spaceflight on muscle. Now, an interesting thing about this particular experiment is not only did we measure force, but we also did gene expression in the same animals. And so, for example, as shown in red there, one of the potential causes for this decreased strength could be alterations in neuromuscular handling of acetylcholine at the synapse. And if you look at the expression changes on the right in terms of the red and the blue, you'll notice that we can also identify transcription factors which may be regulating particular gene expression changes, and these gene expression changes have been clustered together in modules, where these modules are something that's fairly unique to C. elegans because the C. elegans community has a self-organizing map that explains co-changes in gene expression for up to 20,000 different types of experiments. Okay, so cytoskeletal genes were one of the sets of genes that change in response to spaceflight. But the other major set of genes that changed in response to spaceflight were metabolic genes, largely mitochondrial genes. So one thing that we can do on Earth is we can take a worm, as shown in the control. That's a worm displaying calcium levels just as it sits there. And on the bottom is another worm showing calcium levels where you can see they're much higher. And the reason for that is because we've poisoned the mitochondria using a mitochondrial poison. Now, in result of that mitochondrial poisoning, not only do we see elevation in those calcium levels, but as shown on the right, we see a collapse of the sarcomeres, which suggests that mitochondrial dysfunction alone can cause sarcomere disruption. And we've subsequently shown that a drug that improves mitochondrial function can reverse this. So taking these results and thinking about how to translate them into people, what we can do is we can take people, and rather than putting them in space flight, we can subject their muscles to disuse. And so if we look at the bottom in red, we'll notice that there's a decrease in mitochondrial organization in humans who've been subject to disuse. But if you notice, there's a bunch of gray boxes there, which means that there are actually no functional consequences in the muscle associated with that mitochondrial disorganization. Whereas if we look at the top, in green, we see that there are changes in voltage-gated calcium channels, and these correlate with changes in lean muscle mass, which basically means that those mitochondrial organization changes are probably occurring subsequent to changes in ion handling, where the ion handling is the thing that's really causing the changes in the muscle. And if we look at another human study, where we've done bed rest rather than cast immobilization, what we see is the calcium accumulation within the mitochondria seems to be driving the changes in metabolism that we see with bed rest. And as a final example of trying to translate results from worms to astronauts, here's a study where we knew that spaceflight induced changes in gene expression linked to insulin, and so therefore we looked whether we saw similar changes in rodents across tissues and in two cohorts of astronauts. Unsurprisingly, we found that just like in worms, changes in insulin signaling do occur in spaceflight and may be a major contributor to the overall changes in health in response to spaceflight. I hope you've enjoyed the presentation, and I hope you see that there's multiple ways that you can use worms to understand biological changes in response to spaceflight.